next to where you turn them in. Or, no, I guess I didn't put the thing to turn them in. It'll be in a week. Yeah, I don't remember if I set a due date or not right now off the top of my head. I know that's not really helpful, but... Does anybody remember where we left off exactly last time on photosynthesis? That looks about like the right spot where we left off on photosynthesis, I believe. Do you have a place where you ended typing, Emily? Oh, that's right. Or I guess this was Wednesday of last week. No, maybe it was Friday afterwards. Okay. Well, I'm pretty sure that is where we start or left off last time. So we talked about that the leaves have different chlorophyll pigments in there, that there's chlorophyll A, chlorophyll B in the carotenoids, and that the shades of yellow and orange that you see in the fall on the leaves are when the other chlorophylls are dried up, and then you're able to see those spectrums coming out in there. One of the things that the carotenoids can do is provide photoprotection. They're going to help absorb and dissipate excessive light that might otherwise damage the leaves. So the leaves can actually be burned by the sunlight in there when there is too much of it, and this helps protect leaves from getting sunburned in there. So what is going to happen is the chlorophyll is going to be excited by the light. It's going to absorb a photon of light, which is going to come in as energy in there. And it's going to take one of the electrons that is in there, and it's going to excite it up to another level. So normally those electrons like to be in their normal orbits in there, and what it does is it gives it a little bit too much energy temporarily, so it hops into another orbit in there. And so the ground state is when it's in its normal orbit. The excited state is when it's in the higher energy state, when it's hopped out of its normal orbit into the higher state in there. And it's just going to be a temporary situation when it does that, but there is energy involved that it gets from the light to move to that higher orbit, and when it comes back down from that higher orbit to the ground state, there's going to be energy released in there. And that's how it's going to harness energy from the light. So only the only photons that are going to be absorbed are going to be those whose energy is exactly equal to the difference in there. So if it's got too much energy, it's not going to excite it. If it doesn't have enough, it's not going to excite it. It has to be an exact match. So by having more types of chlorophyll in there, that's more potential matches of energy that you can have get excited in there. So some pigments in isolation will release heat and emit light. So the afterglow or the fluorescence is going to be the light that it emits as the electron drops down. So when you see something that fluoresces in the dark, that's what's going on, is you're seeing that energy be released as that orbital goes, or that electron goes from that excited orbital back to its normal one in there. Let's get this a little more centered in there. So in plants, when we talk about the photosystem, it's going to be the reaction center complex associated with its light harvesting complexes. So you've got a reaction center that's your photosystem that's going to be surrounded by these various pigments, which are your light harvesting complexes. And when we do this, we have electrons move, so we have to have an electron acceptor. And your primary electron acceptor is going to be the molecule that's capable of accepting the electrons. And just like with the metabolism reactions where they're redox reactions, that's the same thing that's going to be going on here. You're going to have whatever accepts the electron becoming reduced in there. So when the chlorophyll A gets excited like that, it's going to use the energy to boost an electron, and then it transfers it to a primary electron center. Thank you. So there's two types of photosystems in plants, and they use slightly different wavelengths of light in there. One's going to be 680 nanometers, the other one is 700 nanometers in there. And they just call them photosystem 1 and photosystem 2. They were named in the order that they were found or discovered. That's the only significance to the numbers in there. So the electron flow in there is linear. It's going to flow through the photosystems and other components of the thylakoid membranes. So this is kind of like an electron transport chain that starts things out where you're moving electrons, and what got them started was the energy from the light that kicked it up into another orbital, and then you're just passing it down a chain is essentially what's going on here. So the photon strikes the pigment and boosts the electron. And when it falls down, it ends up exciting another electron with that energy that's going to go in until it reaches the photosystem, the P680 in photosystem 2. So some plants are going to use just one of the photosystems. Others are going to use both, and you excite one and it comes down, and then you excite the other one and it comes down. You could think of that kind of like being where you had NADH come in and FADH come into the electron transport chain in there. It's just two things contributing electrons in there. So it's going to move its electron to the primary electron acceptor. When it does that, it's going to end up splitting water into two electrons, two hydrogens, and oxygens. And it's going to get passed down through an electron transport chain. This electron transport chain is going to use different carrier molecules than the one in animal cells does. So there are the cytochromes in there, the plastoquinone and plastocyanin in there. And then when the electron passes down the chain and finally comes back down to its normal energy state in there, it's going to provide the energy to make ATP. So it's going to provide that proton gradient in there where you've got protons and electrons separated again. And by letting them come together, that's going to give you your energy for making ATP. So it will get passed down to a second electron transport chain through the proton gradient. You've got ferredoxin in there. It's going to do that. And then you're going to have the NADP plus reductase catalase transfer electrons in there. And by moving these electrons and catalyzing that, you're going to have the energy available to go into the Calvin cycle. What do you really need to know about this? You want to know what those photosystems are that you've got the P680 and the P700 in there is important to know. That it's the electrons moving between the excited and the ground state that is generating the energy in there, and that they're getting passed down the chain in there. You're separating positives and negatives again in order to have the energy. It's those same key concepts that are going to create energy here to make ATP that we had in the electron transport chain. So where I mentioned some use just one photosystem and some use two. With cyclic electron flow, this is going to use photosystem 1, but not photosystem 2. So with that one, the electron comes back to its original source in there. When you have the two photosystems, the electron ends up somewhere else. When it comes back to the original source, you don't have to continually have a supply of electrons. It's just a self-feeding cycle that's kind of self-contained in there. When it goes to another source in there, you have to have an additional source of electrons that comes in. Yeah, with photosystem 2, it ends up somewhere else and you just have to keep refeeding it. So it's not just plants that use this, but some of the photosynthetic bacteria in here. And that's where you're going to see most of this with the having just one photosystem and the cyclic. The other one is sometimes referred to as non-cyclic electron flow in there. So the chemiosmosis in the chloroplast versus the mitochondria, both are going to generate ATP by having the chemiosmosis, the separation of those positive and negative charges and ions in there. Some electron carriers are going to be similar, some of the cytochromes, some of them are going to be different. The ATP synthase complex is similar. It's a similar enzyme that's used to phosphorylate ADP to make ATP. The source of electrons in mitochondria comes from organic molecules. It's going to come from water in the chloroplasts. So organic molecules, we mean the sugars that we looked at. The mitochondria are going to use chemiosmosis to transfer the chemical energy from food to ATP. Chloroplasts use the light energy to make ATP in there. And then in the mitochondria, your inner membrane is going to pump hydrogen from the matrix to the inner membrane space in there. In the chloroplast, the hydrogen is pumped from the stroma into the thylakoid space. They are essentially both moving it into a space created by a membrane in there. So they work very similar. If there was, I know that for what you guys are going into with nursing, the whole thing with the photosynthesis is something that is, you probably will never use it outside of a biology class. So if you can kind of look at photosynthesis and think of it as a way to maybe understand the other animal metabolism more, some of it will stick in there. So like if you can look at it, this part in here is probably like the most useful thing, because you would look at it and go back to the other parts of metabolism and maybe another couple things in there would click. Because all of metabolism and photosynthesis is not going to click for you this first time through. If you're looking at it going, I feel like someone put my brain in a blender for this, that's very normal. Everybody feels that way the first time with it. So don't be too concerned about that. So your Kelvin cycle is going to be where you use this ATP and NADPH to take your carbon dioxide from the air and glue it together to make sugars in there. So it's like the citric acid cycle, except kind of a reverse of it. In the citric acid cycle, you're pulling off the carbons to make carbon dioxide from your sugar. Here, you're gluing them back on. So the citric acid cycle is considered catabolic because it breaks something down. The Kelvin cycle is considered anabolic because it's going to build something. So one of the sugars that we had that started out in glycolysis was glyceraldehyde 3-phosphate. It was one of the 3 carbon sugars where we had split things in half. What the Kelvin cycle is going to do is it is going to create this glyceraldehyde 3-phosphate, this 3 carbon sugar. And when you make enough of those, then you glue those 3 carbon sugars together in there. And that's like taking and doing a reverse of glycolysis in there. So we have 3 phases that are involved with carbon fixation. I put the pictures of these in there in Canvas for you, so if you want to look at it, you can. But I'm not worried about you knowing intermediates of photosynthesis at all in there. So you're going to incorporate your carbon dioxides one at a time for the carbon fixation. You've got this 5-carbon sugar ribulose bisphosphate that they abbreviate as RUBP. And it's going to be catalyzed by carboxylase, or they sometimes call the enzyme Rubisco. And why they didn't stick to the enzyme name ending with ASE to make it consistent with things, I don't know. Because Rubisco doesn't sound like an enzyme name. And what it's going to do is it's going to form this 6-carbon intermediate that immediately is going to split into 2 3-carbon sugars in there. So you have taken this 5-carbon piece, glued another one on there to make 6 carbons, and then split it in half. So in phase 2, the reduction, you're going to take each of those 3 phosphoglycerates in there, and it's going to receive ATP in there to make 1,3-bisphosphoglycerate, and then it loses the phosphate to become glucose 3-phosphate. So again, you're modifying it with putting some ATP on there with the energy from the phosphates. And for every 3 carbon dioxides, you're going to end up with 6 of the glucose 3 or the glyceraldehyde 3-phosphates. But only one of them is a net gain of carbohydrate because you're adding into that existing cycle in there. So you begin with 15 carbons in there that have 3 molecules, 5 carbons each. Then you end up with an 18 carbon set in there. Each of them you have 6 of them that are 3 carbons each in there in the end. So you're just rearranging them a little bit to form that. And what exits is going to be used by the plant and recycled to make additional 3 RUBPs in there. So where you can have some of those sugars be used to make structures from the Krebs cycle and glycolysis, the plant can do the same thing. So when you're looking at the plant structure, plants are largely made of sugar, starches in there like cellulose that makes up the wall. This is how it makes those products, those sugars that go into making the cellulose. So then in phase 3, you've got to regenerate the carbon dioxide and RUBP back in there again. So here is where you're going to accept another carbon dioxide in there, things get rearranged, and you end up with your starting materials again so that you can repeat the cycle in there. So your Calvin cycle is going to consume 9 ATP and 6 NADPHs to make those sugars in there. And then it has to rely on those light reactions to regenerate the ATP and the NADPH in there. So stuff that I'm sure is going to seem even less interesting to you with the plants. Sometimes the plants have to make some adaptations off of this normal photosynthesis cycle in there to deal with their particular climates. So in an arid climate, a plant is going to lose water in there. It's going to go out through the stomata, as well as it's going to allow carbon dioxide in. We normally want to let the carbon dioxide in that opening, but if a plant loses water, it doesn't have that electron source in there. So what they'll do on hot days is they will actually close the stomata, but that closes off their source of carbon dioxide in there. So normally they would not be able to get in the carbon dioxide they need for food to build those sugars when you do that. So what some of them will do with that is they call them the C3 plants in there versus the C4 plants in there. You're going to start to have oxygen build up in there, and that's going to be a wasteful process for the plant. So one way of dealing with it is to separate the times that you do these things So what they will do is they can end up taking in the carbon dioxide at night. They'll do the light-dependent reactions during the day, and they will store up the ATP, and then they'll do the Calvin cycle where they need to take in the carbon dioxide at night, is one way of dealing with that in there. Another way that they can do that is to separate things in there. And I'm just kind of summarizing this rather than going through all the details in the notes. You'll have this stuff that you can review back on, but I don't think you're going to really want that much detail in there. On here is to separate where they're going to do these things. And that's where the C4 plants come in there, is they've got these bundle sheath cells in there that are going to be around the veins of the leaf. And then you've got the mesophyll cells that are arranged between the bundle sheath and the leaf. And it creates separate compartments in there so that you can be doing the Calvin cycle in one compartment and the light reactions in another compartment. That way you don't have to be losing your water in the same way in there. So there's sugarcane does this, corn, there's several thousand species of cells that will separate where they do these reactions. It's kind of like if you have a prokaryotic cell versus a eukaryotic cell, where in the eukaryotic cell you do all of these things in the mitochondria. Between the mitochondria and the cytoplasm, you get to separate where the reactions occur, is how animal cells have dealt with it. If you're a prokaryotic bacteria, you haven't separated. It all has to be in the same area. And plants have evolved to do the same thing, where some of them are going to separate where they do these reactions, and others are going to have to keep them all mixed together in there. So one of the enzymes that they can do this to that can help with this process is the phosphoenol pyruvate carboxylase. It will add the carbon dioxide to the phosphoenol pyruvate. It puts it onto an intermediate in there that's got a higher affinity for the carbon dioxide than the Rubisco, and it's going to be more efficient when the carbon dioxide is reduced. So it will help to do this by having something that's going to have a higher affinity for it to pull it away from that cycle until it doesn't need to use it for that cycle in there. And then with the cam plants, this comes from the name crassulacean acid metabolism in there. We call them cam plants in here. So a lot of the succulents, cactuses, pineapples are going to rely on these mechanisms in here to deal with that. So what these are going to do is they're going to open their stomata at night and close them during the day. That way they don't lose water. So if you've seen succulent plants, there's little hen and chicks ones that you see that are kind of pokey. Succulents feel really rubbery. We have them here more so than the cactuses. You'll notice that their skin almost feels more waxy in there to help them protect from the water loss in there, and also allow them to be able to close things off during the day. And they also store a lot of water in them with the succulents in there. So the importance of photosynthesis. The green cells are the only autotrophic part of the plant. The rest of it is going to rely on organic molecules to come from other places. So the only part that's doing the photosynthesis is what's green. So if you've got sticks on the plant that are brown, they're not doing photosynthesis. The roots underground are not doing photosynthesis. They're going to have to rely on other parts of the plant to do the photosynthesis and pass the nutrients down to them. Some are going to form sucrose that's going to be used in other metabolic pathways in the cell. It's going to be used as a building block for other things. Some are going to form glucose that they end up linking into cellulose, which is the most abundant of the organic molecules in plants. And then they can stockpile the extra sugars as starch. So some of your plants that are more starchy, like potatoes, what the potato actually is that stockpiled extra sugar in there. So that is the end of photosynthesis in there.